Thank you very much for your attendance. So I'm going to talk about nanostructured-based Labonchip devices for optical and electrical detection. I would like to start my talk by bringing few examples of engineered devices which has greatly influenced biology and modern medicine. In modern medicine and biology, we're always looking after studying the influence at molecular level. So there's a huge challenge to probe these biological system with the minimum influence. Let's say, for example, in a commercial device like a DNA sequencing machine, which is based on linearization of DNA for genetic studies, breaking DNA by the time we're linearizing DNA, breaking DNA is a bad influence. Or let's say in case of biosensor, which is the best example, is glucometer. Lack of sensitivity, lack of selectivity is a bad influence. That's why there's a huge challenge to bring up with new idea and new devices to overcome these challenges. During my research, I actually tried to work on two different directions and two important challenges. One of them is long-read genomic DNA. But why this becomes important? In today's medicine and biology study, in order to study disease such as cancer, which is a genetic disease, we need to look at, one way is to look at the DNA from individuals. So if we consider a human cell, every single cell consists of like 2-meter long DNA in extended form, which is like 3 gigabase per DNA. So there's a huge challenge to extract these 2-meter long DNA from a cell and extend it on a surface for optical or chemical characterization. Such this technology has a great and very huge application in genomic analysis and in cancer, prenatal diagnosis, in agriculture, animal studies, everything related to DNA and cell. And in order to work on this technology, the method that we are actually using is study DNA under confinement, which I will go through in the next slide. The other challenge that I tried to address during my research is rapid and one-step detection of a biomolecule in real sample. So as I said, in the very good example of a biosensor is glucometer, which is fast and it's already like is an automatic and digital detection method that we can rapidly understand the level of glucose. But we don't have any other method that can compete with glucometer. Nowadays, the method for detection for diagnosis is like a method like ELISA, which takes several days of work and professional technician, but we're always trying to reduce this amount of time and have a rapid detection method by bringing the basically multi-step work to a Labonchip device or kind of single cell step detection method that we are working on in Dr. Kelly's lab at University of Toronto. So this technology again has a great application in environmental monitoring, for medical diagnosis, food safety and agriculture, and there are different type of sensors that has been reported so far based on electrochemical or electrical detection, optical based sensor, or other type. So based on this introduction, I would basically go through, I will divide my talk into three basically parts. First, I will study the first challenge for long-read DNA molecule and how by designing a Labonchip device and a technique that we developed at McGill University, we could basically read a long human genome DNA. And then I will introduce you to a technology that we have patented. This technology is undergoing at McGill, which is for high throughput DNA confinement, and then I will talk about the biosensing and how nanostructuring can affect biosensing, which which I'm following at Dr. Kelly's lab, and it was the essence work of my PhD. So, just a very quick introduction of what is DNA, studying DNA under confinement. So far, there are existing technology to study DNA under confinement. One way is to basically linearize DNA inside the nanofluidic channel. Basically, if you consider DNA as a polymer physics, as a basically polymer molecule, we can load DNA by the pressure gradient into the nanochannels, and by storing contour through basically deflection with the channel wall, the DNA will align through the channels. This method, it's one of basically Dr. Walter Reisner, my supervisor at McGill. He's one of the pioneers in developing nanofluidic technology for DNA linearization. And I had the privilege to work with Dr. Sabrina Leslie at McGill to develop another technology, which is again based on loading DNA into nanochannel, but here we use a convex lens to load DNA, and it's a top loading confinement. So in nanofluidic based confinement, we need for sure a nanofluidic device. So in order to make this nanofluidic device, we can basically pattern nano and micro features on a substrate, and by basically bonding this substrate with a cover slip, we can have these fluidic channels. But in terms of, let's say, fused silica as a material that has a very great application in optical, especially detection method, we need to do RCA and Piranha cleaning to activate the surface, and basically diffusion bonding at very high temperature, like 1000 degrees. So that's why making these devices becomes very challenging in terms of like clean room job and fabrication method. And especially because of the very high hydraulic resistance that's introduced by the nano channels here, loading DNA into the nano channels through the, from a micro scale environment cause breakage of DNA. That's why in terms of loading long DNA, which is like human DNA, it's very challenging to load a long human DNA into nanochannels, especially when it comes to channels less than 100 nanometer, which according to polymer physics, this is, in order to have the high extension and full extension of DNA, we need to go to channels less than 50 nanometer. That's why we can see that how challenging this technology would be. So in basically click technology, which we developed at McGill, the action of basically this instrument is based on the action of a convex lens. By using a convex lens, we can basically deform a cover slip over a gap and create a variable and tunable confinement that can load and drive DNA into the nanochannel from top. This method has the advantage of basically open system. That's why the loading DNA happens very gradually, and we avoid breaking of DNA during loading process. And again, because this is an open system, and we only use a double-sided tape to bond the cover slip with the nanopattern channel, the nanopatterned device. That's why we don't have the challenge during fabrication process. This method, by using this method, we could successfully confine DNA molecules into channels less than 25 nanometer. So, which is a great success. In terms of lambda DNA, which is 48 kilobase per, we could reach the full extension, which is about 20 micron. And according to the fluorescent microscope, we could actually see that the full extension of lambda has achieved. And in terms of fabrication, just quick basically introduction that in order to fabricate the nanochannel devices, we use e-beam lithography technique, but when it comes to channels less than 100 nanometer, it becomes again challenging because we need to basically have an optimum condition between the e-beam exposure dose and ZEP resistant layer, which is the layer, the polymeric layer, which we use for nanofabrication method. That's why I'm just repeating this basically challenging process, and I want to say how challenging it would be to make the nano channels less than 100 nanometer to basically introduce you to the next step of the technology. After successful confinement of DNA in nanochannels using click technique, we decided to go for the next step. I was talking about human DNA, genetic DNA. Human, which is like super long, it's more than like lambda DNA, which is only 48 kilobase per. Human DNA is almost, it's 3 gigabase per. So there's a huge challenge to load all the DNA coming from single cell into a substrate. So we decided to use click method and taking advantage of this open system to load long DNA inside the nanochannel. But here, again, we need to actually extract this DNA from a single cell, because every time if we do this besides single cell, we need to do this on chip. Every time we extract DNA off chip, by loading the DNA into the device, part of the DNA will break, and this is a bad influence that we need to avoid. That's why we need to design a device where we can extract DNA on chip from a cell. That's why, again, we need part of the design, we need a cavity in our device that we can trap the single cell. And again, we need a method where we can basically lyse this cell on chip. That's why we have challenges for trapping single cell, for bringing the lysis buffer into the device, extract DNA on chip, and then loading the DNA into the nanochannel part. Our device is basically 1 inch in size, and it's made of fused silica, and it's the center of the fused silica basically is patterned with the microcavity for trapping the cell, and we have post arrays to help untangle DNA, and we have nanochannels to confine DNA for optical observation, and the microchannel to bring the fluidic and lysis buffer into the device. Here, because we are dealing with single cell, we need to bring lysis buffer so we can lyse the cell and extract DNA on chip. In click technology, basically we have a huge hydraulic resist when the push length is all the way down. That's why we need to overcome this problem by having a suction through hole close to the confinement system. Here, when the lid is all the way down, we have a very high resistance, and we cannot easily actuate flow for cell lysis and other buffer exchange for other biological exchange. That's why we design a suction through hole close to the confinement area and by applying a negative pressure, we can bring enough solution into the device and initiate the lysis. That's why, again, making such this device adds basically more step into the fabrication, and fabrication of this device might take more than usual. So this is how the device basically performs. By using the click basically push lens, we can trap one single cell in the cavity that we designed, and by actuating the flow through applying the negative pressure from the suction hole, we can bring enough lysis buffer and we can initiate the basically lysis as soon as the lysis buffer introduced. Here we can show that less than 10 minutes the lysis basically begin, which is a very good record in terms of like a rapid process. And in less than, in almost like 30 minutes, we can have all the chromatin digested and the protein digested, and we can have pure DNA extracted inside the confinement area. By basically using this technique, we can confine super long human DNA inside the nanochannel. This is almost 1.5 megabase per human DNA, which is the longest that has been reported so far. And we can have like a genomic DNA with different size, like kilobase per, megabase per. And this is one of the problems that we had, that lack of uniformity in terms of like confinement. But the next step would be how we can basically get the genetic information out of these optical observation. We observe DNA in the nanochannel, extended DNA, but how we can get the genetic information from the extended DNA. But before going to that discussion, I would like just to introduce DNA more detail. If you look at the basically DNA double helix, it's composed of 4 basically important elements. C and G abbreviation are always bonded together. A and T are always bonded together, and each we call a base pair. So as you can see here, C and G has like 3 hydrogen bonding. However, A and T has like 2 hydrogen bonding. That's why we assume that we have more strong bonding here comparing to A and T bonding. This is basically, this knowledge helped my supervisor, Professor Reisner, to develop with an idea by the time he was postdoc to basically patent a technology named melting mapping, which helps basically to study and map the DNA and study DNA in terms of like genetic information. So by using the melting mapping technique, which is just simply by increasing the temperature, we can locally melt DNA from the part which is rich in 80 base pairs. So in this case, for example, in terms of lambda DNA, when we look at the fluorescent image, we can see a black and white pattern. And the reason is, by the time that this 80 rich part is melted, the fluorescent dyes, which is bind to the DNA, will release. That's why we cannot have any fluorescent basically distribution here. And we can have a black and white image coming from basically this DNA. And this is how basically by using some coding, we can understand what is exactly the pattern of DNA, and we can relate it to, by bringing like bioinformatic information and coding, we can relate it to these basically mapping of DNA. We could so far do it for lambda DNA. We haven't done it for genetic, like human DNA, but this is something that we would like to pursue for human DNA as well. So the first part of discussion was introducing a Labonchips device where we can do, and basically we can perform an on-site, on-chip process for trapping a single cell, extracting DNA, and linearization of DNA inside the nanochannel. Everything was done on chip. So we had like achievements here, but what we cannot achieve, let's say, so far, helped us to come up with a new idea for developing our technology. That's the second part of my discussion. The approach to overcome this difficulty was to introduce an electrode close to the confinement system. So we tried this method as well, and by introducing voltage and electrophoresis, we could bring more DNA into the confinement area, and we could have a high throughput device. But again, the problem of ununiformity and the problem of buffer exchange is still there. And another issue that I can tell you, we use a 1-inch device, but in order to have a DNA confinement, we can only confine DNA in less than a 1 micron space inside the device. So it means that we have a 1-inch device, but we can not have nanochannels everywhere to basically confine DNA all over the channels. That's why we come up with another technique, which I call it DP. I'm so sorry. I call it DP confinement. In this technique, we simply use a dielectrophoretic phenomena to load DNA inside the nanochannel. Here, by basically applying voltage and adjusting the frequency, we can load DNA inside the nanochannel, and again, we take advantage of an open system. We don't have any bonding here, and the only thing is like choosing basically smart material like ITO, which act as our electrode to generate the field, and the silicon nitride to just separate these channels and create the nanochannels or microchannel or any feature we want. So the technology is very simple, but the result is really amazing. We can have like a high throughput DNA confinement inside the nanochannel by just action of an electric field. So again, here we can have high throughput. We have the advantage of open system, and we don't need to go through a difficult and challenging fabrication. So the only basically the physics behind this phenomena, which I'm sure you all know better than me, it's dielectrophoretic, which is based on the formula for the dielectrophoresis. And here, in order to understand this phenomena, we just need to compare a dielectric particle in uniform and non-uniform field. A dielectric particle, if it's happened to place in a uniform field, the Coulomb bar between each side of the particle in magnitude is the same, but the direction is different. That's why the net force becomes 0, and we cannot have any movement of the particle. But when the particle happens to be in a non-uniform electric field, the net force will be like either positive or in a direction of the field, or a positive direction of the field against the field. This is what we call the dielectrophoretic movement, and we can use basically these phenomena to confine DNA inside our nanochannel and control the movement of DNA. So using this technology, we can actually we can confine DNA molecules as a dielectric particles from a microscale confinement into almost a 10 micron size confinement, which means that by using the dielectrophoretic without having a microscale, microfluidic channel, we can actually confine DNA. We can accumulate DNA in a microscale confinement, in a microscale basically confinement as we have here. So by using a COMSOL simulation technique, we could predict the position of the DNA dielectric particles on the base electrode that we designed in the system, and we can see that even our result in terms of accumulation of DNA on the electrode is in a very good adjustment with the simulation result that we have here. After confining DNA into microscale, we tried the nanoscale confinement, and as we can see here, even in the nanoscale confinement, we can load and unload DNA into the nanochannel. This is what we call a tunable confinement. So this is somehow like the action of our push lens click for confining the DNA, but here we don't have any mechanical component for pushing the basically cover slip. It's just the action of the voltage and frequency for loading DNA. And another interesting result we could see here, that's why I was insisting of talking about the nanochannel fabrication. Here we can actually confine DNA into 100 nanometer channel, and we can reach full extension of lambda DNA confinement in even 100 nanometer channels. So by using this method, we don't even need to go to channels less than 100 nanometers. So again, we can skip the fabrication challenges in fabrication of nanometer channels. So what we can actually achieve from this part is we introduced a technology based on the nanostructured silicon nitride ITO. We could establish a baseline fabrication method for this method in terms of material characterization. We could successfully confine DNA into 100, 300 nanometer and 1 micron nanogrooves, and we could reach high throughput confinement, and we could develop a method for explaining the confinement in our device. So I finished my talk about the Labonchips devices and the DEP technology for confining and DNA, and I would basically start the discussion about nanostructured electrodes for non-enzymatic and direct biosensing, which basically the essence of the work I did during my PhD and now during my postdoc at U of T. And basically here, first, I will talk an electrochemical detection method, which is the method that we use for biosensing. And then we'll talk about the nanostructured electrode and then detection of dopamine, glucose, and DNA-based sensors. This is the second challenge that I will address in my research. So in terms of electrochemical detection, we can just simply use a very electrochemical cell, which has 3 components: working electrode, our counter electrode, and reference electrode, and a electrochemical working station which attached to a processor. Nowadays, we can have all this set up in a very small chip. This is something that we are working on right now. And basically in this method, what is important is the working electrode. So this working electrode, so far in most of the electrochemical method, is from gold surface, because we can have like many reaction happens on the gold, and in terms of DNA-based biosensor, we can easily probe DNA by a thiolated thiol bonding, and we can have a DNA probe on the surface of the gold, and by having the target DNA and having the hybridization on the surface, we can have an electrical signal. Even we can have a biomolecule, basically reaction, biomolecules like glucose, dopamine, ascorbic acid, uric acid, they all undergo electrochemical reaction on the surface of gold or other catalytic material. And we can have an electrochemical signal, what we call, that can show us if the target molecule is existed in the solution or not. So this is how basically we analyze, this is the method we use for our biosensing. But since I'm a material engineer, so the part that I'm very much interested is designing a working electrode that can be more efficient and sensitive. So I very much believe in nanostructuring as a material engineer, and I believe that by nanostructuring we can solve all the problems in the world. That's why I try to design electrodes which are efficient in terms of sensitivity and limit of detection in real sample, real condition. So the essence of my work, I did my PhD at Sharif University, and there I was working on a special nanostructured electrode based on nanotubes, and I was doing a collaborative work with my sister, actually she's also here, and during my PhD, and we developed a nanowire structure for biosensing. And now at University of Toronto, we're working on a nanostructured microelectrodes, which is an on-chip method for biosensing, which has great application in basically in terms of like real sample detection, high sensitivity, and of course in later on for bringing into like a digital basically sample digital sensing method like we have for glucometer. So first, talking about TiO2 nanotubes, it has great advantage in terms of large surface area, and because it is like a semiconductive material, we can actually modify the surface of these electrodes with variety of material, like catalytic material like palladium, platinum, gold, quantum dots, and other kind of oxide. That's why we can make a variety of composite material actually to develop our electrode and get different application. So, and another advantage of this electrode, again, we are an engineer, so we always try to find simple methods. So we can easily make these material by basically anodization of titanium sheet, which has again advantage of in terms of fabrication. So the first electrode I proposed was a composition of palladium nanoparticles and TiO2, and I used this electrode for detection of dopamine in the solution. Dopamine is one of the biomolecules that lack of dopamine can cause like schizophrenia, Parkinson's disease. That's why we need to detect dopamine in the presence of other molecules with a very low limit of detection. Here in the first experiment, we could see that the electrode can detect dopamine in quite high concentration micrometer range, but we couldn't see a stable, actually we couldn't get a very stable detection and signal in low concentration of dopamine. And in terms of stability, PD-TiO2 electrode didn't show a very high stability. Surprisingly, by adding platinum as a second catalytic material, we could have a linear range of detection in even low concentration of dopamine, and we could reach a high stability in terms of the electrode life even after like 100 cycles. But the problem here we have, because as I said, in real condition, dopamine is not alone, is in coexistence with other biomolecules like ascorbic acid, uric acid, or other type of biomolecules. That's why one of the characteristics of the sensor is the selectivity. Here our electrode can show selectivity in like low voltage pH, but not in neutral pH, which is close to basically human body. That's why we try to introduce another part into another component to our electrode, and we developed the AUPT-PD TiO2 electrode, and in this electrode we could reach a very high sensitivity in terms of detection of dopamine. And the important part here was we can have relatively higher selectivity of dopamine in presence of ascorbic acid and uric acid even at neutral pH. This electrode was successfully used in a real sample, human urine sample, with a very good acceptable, let's say, because the paper is for 2011. By that time, it was a very acceptable range for detection of dopamine in the real sample condition. Another electrode that we developed, we introduced carbon into our system, and by introducing carbon, we could have a very high conductivity in terms of detection of dopamine and uric acid comparing to the metal-based electrode. And beside detection, we could actually characterize different concentration of dopamine and uric acid in their coexistence. It means that we could reach multiplexing here, which is one of the again properties of the nowadays sensor. We can characterize dopamine in a fixed amount of uric acid, and we can characterize uric acid in the fixed amount of dopamine. So the second, study is selectivity, which we can show that because of the high conductivity of the electrode, our electrode shows a very good selectivity in terms of detection of ascorbic acid in presence of other biomolecules. And the other study that we did was the nanowire-based metallic electrode, which we actually use this electrode for detection of glucose, and we could actually reach a high sensitivity of detection of glucose. None of these electrodes has been commercialized. These are all the alternate materials that we can introduce that later on can be, let's say, used in on-chip biosensor, or let's say commercialized devices in terms of like biosensor. The second part basically of my research, which I'm pursuing now, it's on-chip nanostructured microelectrodes. In this method, which has been developed in Dr. Kelly's lab since 2009, the electrode, the sensor platform, is a gold nanostructures, which is electrode deposited in a microscale aperture. So by using these basically spiky shaped nanostructure, we can have a very high sensitivity in terms of DNA hybridization. When I say DNA hybridization, it's the essence of the DNA-based biosensor. I don't want to go into more detail in terms of DNA-based biosensor, but what I can say here is based on DNA hybridization. So what is important here is we need to have a probe of DNA on the basically attached to the surface, and the target DNA needs to basically hybridize with the probe DNA on the surface. That's why functionalization of the surface with the probe DNA becomes important. As much as we have more probe DNA on the surface, we can have a higher signal. In Kelly's lab, basically, they proved that by nanostructuring an angled surface, we can have the maximum density of the probe on the surface. So by the time I joined Kelly's lab, I tried to basically develop try different surface and different structures to see which of them can basically give us a high efficiency in terms of DNA hybridization. And later on, we can see that in different assays, sometimes we avoid DNA hybridization. That's why it's important to understand our surface and how to control with the surface. That's why this is the And that's why I tried the position of these structures at different voltage and different conditions. And in order to basically control our condition, we need to model. Hopefully, there are already established electrochemical models for nucleation and growth of electrodeposited material. That's why we adopted these models, and we can predict, basically, we can explain our deposition condition. For example, for a bulky structure which happens at a positive voltage, we can have like a nucleation all over the aperture, because nucleation happens with a progressive control, and in the spiky shape structure, we can have nucleation just partly in the aperture. That's why the spiky structures happen. And as we go to more negative voltage, we can have more flaky structures, where we can see, later on we can see that these structures can have different efficiency in terms of DNA hybridization. Here, with the bulky structure, we can almost, the rate of DNA hybridization is almost very low. But when it comes to more spiky structure, which happens at 0 up to minus 150 volt, we can have the maximum DNA hybridization. And when we come to more flaky structures, we can even have again less DNA hybridization. We take advantage of these different structures to study one of the assay that it's undergoing in our lab, and the assay developed by my sister again. So we adopted that assay, and we tried these structure, and we can see that even in terms of real detection of an antibody, we can have different efficiency, which again, basically, we can again learn that controlling the structure can even help us to control the efficiency of an assay. So I think I can finish my talk right now, and we can have some time for questions and for answering questions. And so far, what we achieved here again was in terms of nanostructuring and biosensor is by non-enzymatic detection of biomolecules like dopamine and glucose can happen with nanostructured electrode. When we say non-enzymatic, it means that we don't need any ligand or any material, any organic material on the surface of electrode, and the electrode itself can basically we can have the reaction on the surface of the electrode, which is very important in terms of stability. But we can basically reduce the detection limits of dopamine and glucose in nanometer, which is again important in terms of like diagnosis. And we can improve the selectivity of the detection by alloying. And again, by controlling the nucleation and growth, we can have a high surface area of nanostructured electrode, which can basically, and we can have a high efficiency for DNA-based biosensor. But every research, I have done this research for almost like 10 years, like Labonchips, biosensor, nanostructure. So the outcome of the research which I'm following right now as a researcher is that I can have like a technology whereby on-chip analysis, we can do on-chip analysis. On-chip analysis have advantage not only for genetic studies, for biosensor. Nowadays we use like lysis bacteria on-chip, and we can extract the DNA and analyze and detect, let's say, bacteria based on these on-chip analysis. And we can improve sample delivery by DEP. I told you that by DEP we can have a micro-scale confinement by just action of a voltage and frequency. So I believe that by using this technology we can improve sample delivery even for biosensor without using a microfluidic device. So we can improve the biochemical and sensing by nanostructuring material, which is something that we are even doing right now for commercialized device in Dr. Kelly's lab. And we can actually improve the synthesis, and we also improve the synthesis protocols for nanostructured material like nanotube, nanowires, and microelectrodes. At the end, I would like to acknowledge all universities and departments that I have been affiliated with so far. My PhD supervisors in university, Sharif University, and then I had the privilege to work at one of the state key lab in China for less than a year, and my supervisor, Dr. Chai. And at McGill, I was affiliated with a physics department, Dr. Walter Reisner lab, and Dr. Sabrina Leslie lab, and Genome Quebec Innovation Center, Dr. Robert Sladek, and right now at Dr. Kelly's lab, which we have a very big group, and I'm actually working with very different people from different backgrounds now, and we are hopefully we can develop more interesting devices, and I can talk about it in my next talk, let's say. So if there's any question, I would be happy to answer.
Okay, thank you for the great talk. So in the ITO part, I had a few questions there. Is there a specific reason why you use the silicon nitride?
You know, in order to, I could either pattern ITO, which I wanted to avoid because of the problem that fabrication of ITO has in terms of controlling the etching. I use silicon nitride as an insulator layer to just create the electrodes. So if you can pattern ITO, it would be also possible. But you have electrodes everywhere, let's say. We want to avoid confining DNA in other places. We just want to control confining DNA in a specific place, which is just our electrode.
So basically, any insulator can be used?
Sure,
silicon
yeah.
oxide?
I have tried oxide too, which is possible. But silicon nitride was the first choice for other reasons. But it's also possible that you can use silicon oxide as well.
Yeah. Since my background is electrical engineering, I'm really concerned about that part. What is the spacing between the upper electrode and the silicon nitride? Is it important? Is it not?
The spacing is important, but you just need to keep the spacing in a level that you can create the field. And of course, you should avoid connecting the two electrodes. Basically, so the spacing for us was a few microns, but it would be possible to go more than like up to 30 micron, 50 micron.
And are these devices largely used or just in the university lab?
It's which devices? The nanofluidic devices are widely used. The first image I showed you, Nanobiogenomics, it's a commercialized, it's a company that's using these devices, nanofluidic devices, based on for genetic studies, for DNA sequencing. But what we develop at McGill is, in terms of DEP, we have patented that technology, but we haven't commercialized the technology. I hope that we can commercialize that technology too.
Thank you very much. It's a nice talk and it's very dense. The content was very dense. Actually, I have also a couple questions. The first one is, the nanostructured DNA trapping device is reusable or not? After, for example, doing a test, is there any contamination problem or can you clean it or not?
Yeah, that's a very good question. In terms of non-enzymatic detection, for example, dopamine and glucose, as I mentioned, we could use these devices for many times. Of course, when you want to collect results in order to, and you want to trust your results every time you use like different electrodes, so you can have like fresh results, so reliable result. But in terms of stability, we could actually use these electrodes for more than 100 times. And also we could store these electrodes for more than a month and we can still get the same results. But in terms of DNA-based sensors, which is based on the probe on the surface, we cannot use them more than, unless you wash and remove the probes and then you basically functionalize the surface again.
Mm-hmm. Yeah, from this perspective, can you please compare two methods? Like one, the nanostructure grew one, you use the whole DNA sequence and then get some fluorescence images or intensity measurements. In the second one, you functionalize the surface and then you use one strand as a probe and then there is a complementary DNA strand and then you do the same thing. Is there any? Yeah.
Yeah, actually that's again, it all depends on your application. For example, in terms of second approach, which nanostructuring, we can even have genetic studies because we are hybridizing DNA. So if we have like a mutated gene or something that we want to look at, we can have the probe which is complementary to the target, and we can use that probe, DNA-based and probe system, to analyze and to basically detect that gene. In Dr. Kelly's lab, there's a paper which is published in Nature Chemistry, I guess, which they could show that by using nanostructure, they can detect like specific genes as well. But what I talk about in the first basically part of the talk in terms of like DNA sequencing, it's a very wide study. You're not just looking at specific gene. You can basically sequence all over the DNA, which of course has a very different application. If you want to study sort of like genetic disease, or if you want to see, for example, I remember by the time I was at Genome Quebec, the people would come and they ask if because we can analyze the embryo cell, and they want to see if the child is healthy enough. For example, if they have any genetic background in their family, they can find out if the gene is, for example, presented in the child's DNA chromosome or not. So that kind of study has a different application, and the sensor has a different application.
Very short. Okay, if you don't mind, sorry. Again, is there any DNA strand length limitation with comparing again the first method and the second approach? Because in the first one, in a nanostructured groove, you have very long DNA sequence, and in the second one, as I know, oligonucleotides are used, which are very short strand DNAs.
Yeah, exactly. It's only like maximum 25 base pair, because we're talking about probe. And again, I repeat that in the second method, we're looking at a specific genes, let's say. We're looking at a specific target. But in the first method, it's very wide. We're looking at the whole genome of human, animal, bacteria, plants.
And lastly, for the dielectrophoretic study, is there any side effect of heating because of dielectrophoretic? Because you mentioned unmelting DNA sequences also, and I don't know exactly at what temperature DNA gets melted.
Our simulation result doesn't show any heating effect, but I haven't measured the heart of the device. I haven't measured any, let's say, temperature difference. But what I can say that the DNA that we linearize does not melt by applying, unless if you want to go through the melting method, we need to melt, like apply heat from outside. Unless we come up with another, let's say, improvement on the device, I would be happy if I can melt DNA inside the nanotunnel by just applying voltage. So this is something that we can look basically into for the next improvement of the device.
So it would be an advantage if you have such a thing.
Again, it depends on the application. Yeah, if you want to use the melting mapping for mapping DNA, that would be an advantage, and I would like to look at that part too. Yeah.
Thank you for your talk. I have a couple of questions about your melt mapping. So the commercial product with the nanochannels, they will be putting the DNA through and reptating it, like just putting it through linearly, and therefore they can read off the base pairs one by one on the other side, correct? Whereas with your trapping from the top, you don't have that. You can't move the DNA through a channel.
The DNA, you know, the DNA will diffuse inside. You cannot basically keep DNA inside a channel forever. And the DNA will diffuse inside the channels. But as long as the click lens, the push lens is down, you can observe DNA for a long time, as long as the DNA is down. But by the time you will see that the DNA will start diffusing inside the channel and leave the basically observation area. So
right, but this doesn't give you diagnostic information. This just, you can just see that there's DNA
yeah,
there.
that's what yeah, that's correct. By using all of these methods like DP, nanofluidic, we just observe DNA. For the next step, which is melting, which is mapping DNA, getting genetic, basically extracting genetic information, there are other methods. Let's say melting mapping is one of the methods. But the commercialized method to get the genomic information is, for example, labeling DNA. As if you saw the first image, they have like labels of for example, if you're looking at the methylated site on DNA, you can label those methylated sites, and then by basically looking at the positions for different positions, and every company has its own, let's say, method for looking at the DNA. For example, in bio-nanogenomics, they look at the linearized DNA and look at specific part of DNA by labeling. Or for example, there are another technique like Illumina or Pacific Bioscience. They use different technique for sequencing DNA, which is based on trapping DNA in, let's say, for example, Pacific Bio, trapping DNA in a nanopore. And by using a polymerase, they can basically rehybridize DNA frequently and get information. So that's a complete.
But with your method, you need to do something like melt mapping from the top down.
But melting mapping is not commercialized method. Right. So this is just, no, this is only for the research purpose. So, yeah.
With your melt mapping, can you see AT or GC mutations?
Yeah, that's what we're looking at. In melting mapping method, because we know from the basically from the DNA structure, we know that TA is less strong bonding comparing to GC. So when it comes to like a melting point, TA melts like sooner. That's why we have this barcode like black and white pattern. And we can see, but we haven't done it for genomic DNA. We have only done this for specific genome like lambda DNA, T4 DNA that we know the barcode. It's something like X-ray, for example. We know the barcode for this.
Because like a heterozygous mutation would create a hole that you wouldn't know what it was.
Yeah, it's possible.
Thank you for your time.
You're welcome.
I'm thinking about the first technique you showed where you use a lens to compress the space. I'm presuming there's nothing special about it being a lens or optically clear. It's just available, right? And so I'm thinking about a piece of string that's all tangled up. And if you were to compress that piece of string in a confined space, there would be nothing that would make it untangle and line up. Do you see what I'm thinking about? If I had a piece of string that's all tangled up and I compressed it down between two spaces, it would just be more tangled. And so why is it that these pieces untangle and lay in the grooves that you've made?
That's exactly correct, and that's one of the problems we had. That's why we have post arrays between the cavity and the nanochannels. The post arrays help untangling DNA. But again, in order to, because we need to adjust the position of the push lens with respect to the nanochannel in a way that we don't fully confine and basically block the part which the nanochannels are. Because if we have the full confinement on top of the nanochannel, nothing would be there. It's like full confinement. And as you said, if we have like bundled DNA, it will just stick to the channels. Because confinement comes to up to 2 nanometer, which is like very small. That's why we adjust the push lens in a way that we don't have that sticking problem. And we have post arrays to untangle DNA and a flow to basically actuate the flow in a way that the DNA, because the DNA can also be linearized with the action of the flow.
Yeah, thanks. That helps. Can I just ask one more question? How long a piece of DNA can you straighten out like that? Maybe you said it, but I didn't catch it.
So far, we were able to linearize 1.5 megabase per DNA, which is long, but not a very high throughput. Of course, comparing to
In terms of micrometers, how many micrometers?
nanometers.
How many micrometers is that?
Oh, you mean the whole device?
No, 1.5 mega.
Oh, okay. The 1.5 is like three.
It's just a rough estimate.
Yeah, 1.5 gigabase per is 3 field of view. I'm thinking about our field of view. I think it was 100 microns. So maybe like 300 micron, if I'm not mistaken. I just forgot about our field of view. I think it was 100 micron. Roughly 300 micron. Should be.
It's kind of a rookie question about how they deal with the DNA. I don't have too much knowledge, but I have interest in. So, as far as I understood, the DNA diameter is basically sub 10 nanometer. Is that right?
Diameter, yes. Even, yeah, it's in terms of like.
So, but when they map it, you talked about, you confine it there and then you optically analyze it, or?
Yeah, the method that we have here is optical detection, because we look at DNA when it's linearized, and then in order, there's a difference between analyzed DNA and just looking at. So here we have a method that we can linearize DNA and look at the DNA. Okay, but what is the use? You look at the DNA. So one, basically, people look at DNA, like people like Walter Reisner. He look at the DNA extension, compression, like basically polymer physics study. But if you want to look at the genetic side of DNA sequencing, then by looking at DNA, we cannot get anything. We need to label DNA. We need to get that genetic information. So one way would be DNA labeling, which is commercialized now. One way would be this melting mapping. And I'm trying to figure out like another method for mapping DNA, which maybe later on I can talk about when it's developed enough.
No, my question is like, do they basically look at the sequence using an optical microscope? Because that's too small for an optical microscope.
Sure.
Right?
It's a fluorescent microscope. Yeah, optical microscope.
Okay, so basically, the images that you were showing, that it was bright and dark.
Sure.
It's basically the glow from the DNA, depending on where it
Sorry
comes from.
if I didn't, because it was a very
Basic
dense-
question. I know, this is
No,
a
no,
basic
no. It
question.
was very. Yeah, you're right. But I had to talk about this part. We label DNA with fluorescent dye. You know, the DNA itself, it's not bright at all, and it has Brownian motion inside the solution. But in order to see DNA using a fluorescent microscope, we label DNA with a fluorescent dye. For example, in this case, we use yo-yo. Yo-yo is a fluorescent dye which attached to the DNA, nucleotide part of the DNA. That's why we can see a bright, let's say, worm, I say, under the microscope when it comes to linearized DNA. And that's how we can observe it.
And where there is dark, the AT and CG, depending on where the sequencing happens, the darkness and the brightness relates to that. Is that right? Okay. And just one more question related to that. So you were mentioning you'd be happy if you could melt by applying voltage, but can't you just melt by applying the whole, by increasing the heat to the whole thing?
Yeah, of course. But if we're thinking about commercialization, it would be nice if we can, let's say, do it in a more simple way. So if we can, because if we want to apply heat, it means that we need to add another component, let's say a heater. But we want to avoid like complication here.
Can I ask what's the temperature difference between the melting point of AT and the CG?
I can tell you roughly that melting point of DNA, which is about 100, but we cannot go up to 100 because we will damage the objectives of the microscope. That's why we add a chemical like formamide that will reduce the melting temperature up to like 37, 40 degrees, and which is the melting temperature of TA, which is the lower one.
Thank you very much for coming, everybody.
Thank you very much. Great questions.